Hello, I'm Jolien, Associate Director of Commercial Strategy with Teiko Bio. Today, I'd like to show you an example of Teiko's approach to unsupervised clustering and how we used it to find a surprising cell population associated with overall survival in Pisces RADIOHEAD's urothelial carcinoma cohort. At Teiko, we believe that gated and unsupervised clustering analysis complement each other. To start, gated analysis is a great way to cover your bases in your projects. Our panels are designed to capture over 30 key cell populations across the innate and adaptive immune system that are commonly involved in cancer biology processes. Within each of these cell populations, you can gate on individual state markers to get a sense of that cell's functional state. A natural limitation to manual gating, however, is you can really only gate on 2 state markers at any given time, and it is very difficult to find populations with more than 2 markers. Unsupervised clustering analysis is a great way to supplement this gated data. By feeding all immune marker expression data into an algorithm, you can detect between 80 and 200 unique immune cell populations, because unsupervised analysis will evaluate all state markers within a population. However, due to the sheer amount of data, it can be challenging to analyze the results. To show you how unsupervised clustering can yield interesting and unexpected results, I'll talk about a result from our project with the Parker Institute for Cancer Immunotherapy. As part of their RADIOHEAD study, we analyzed peripheral blood mononuclear cells from a cohort of 40 urothelial carcinoma patients that were treated with anti-PD-1. Univariate Cox regression analysis on gated cell populations showed that on-treatment regulatory T-cell frequencies were associated with better overall survival, as is seen here in this volcano plot. Responders were seen to have, indeed, about 2 times higher regulatory T-cell frequencies than non-responders on treatment. This result feels counterintuitive, as regulatory T-cells are usually associated with worse outcomes to immunotherapy. When we looked at individual state marker expression in our gated data, we did not see regulatory T-cells associated populations show up in association with better overall outcomes, meaning we were not able to pin down the regulatory T-cell phenotype associated with better outcomes. To get a better understanding, we split patients into 2 groups based on response and development of an immune-related adverse event. FastPG clustering identified over 80 clusters, in which 2 were mapped to a regulatory T-cell phenotype. Quisquel-Wallace expression analysis showed that responders specifically had 2 to 3 times higher frequencies of regulatory T-cell cluster 2, but not regulatory T-cell cluster 1. You can especially see this over here when comparing the change in frequency from baseline to on treatment in cluster 2. We used UMAP to visualize the differences in regulatory T-cell cluster 1 and 2. We saw that in cluster 2, in green here, had lower expression of CD38, ICOS, and CTLA4, and mixed expression of TIGIT. This marker combination would suggest that responders have specifically higher frequencies of inactivated regulatory T-cells on treatment. This brings us back to how gated and unsupervised analysis complement each other. Teiko found that total regulatory T-cell frequencies were associated with response using gated analysis, but the limitation to individual state markers made it difficult to pin down the exact phenotype responsible for this observation. Unsupervised clustering did find 2 distinct regulatory T-cell clusters and showed that the population associated with response had an inactivated phenotype. While challenging, Teiko's analysis pipeline was able to find this surprising result. Don't hesitate to reach out if you want to learn more at teikobio.com/contact.